Okay, hi everybody. So up to now, you've seen mostly things that live in vector spaces, and now we're going to move to a much more interesting space. We're going to move to graphs, and that's actually where the fun is. As you'll see, you can do different things, you can do better things, you can do worse things, but it's definitely a different world. I have to put these slides, so if I advertise scanners, it's because they pay some of my salary. And here's to start. I just want to give this at the beginning, some useful code that we don't really have time to do a practical in the end, but especially the one on top, the edu ML brain graphs. I put together lots of the techniques we're going to show today, we're going to discuss today are in there, so you can follow. Unfortunately, it's still MATLAB. I'm converting all my stuff to Python and R, but this you can use. Software has a bit more stuff. And then if you want a nice visualization like the brain that you see in the back with this 3D structure in MATLAB, you can use this flexible brain graph visualizer. So yeah, this you can all download, and you can basically see what we're gonna see today. So what are we gonna talk about? First, I'm gonna give you a bit of context. Why do we care about connectivity? This is about clinical neuroscience mostly. I'm gonna show you how you can do decoding, so how you can do machine learning with this kind of data. I'm gonna talk about some challenges because it's even more difficult than with activity or structural data, unfortunately. And then I'll give you some take-home messages. First thing, of course, is we need to know why do we care about connectivity. Well, there's this very nice paper by Fornido et al. That I recommend that essentially shows that many diseases, many things that happen in diseases can be described nicely in a graph framework. Or conversely, if you have some ideas about graph structures, about how things are connected in the brain, then you can also have a nice clinical interpretation. The prime example is diaskisis, this idea that if you have a disconnection between parts, you can actually impact things that are not directly closely located. So in stroke, you have results like this. We also have recently contrary results that show that this does not necessarily happen, but this is nevertheless a very interesting conceptual framework. So in general, I think brain networks are a very good unit to think about the brain. And if you've been coming to OHBM for a while, you know that, of course, we're very interested in this. One small distinction, when I say network, I mean the actual thing in a brain that you can touch, or it could also be functional connectivity, but it's an actual something that has physical relevance. And when I say graph, it's the mathematical abstraction. So I try to be careful not to mix these two terms. What can you do if you have connectivity data? You can do three things. You can do diagnosis, you can do prognosis, and you can do treatment planning. In terms of diagnosis, you've seen lots of examples today of direct diagnosis. So that is, does this person have the disease or not? So direct diagnosis. More interesting is differential diagnosis. So does this person have disease A or disease B, C, D, E that look very much like disease A? In structural imaging, you have lots of examples like this with multiple sclerosis and mimic diseases. In functional connectivity, it's also the case. So many diseases actually will impact the default mode network, but in different ways. And so if you have subtypes, especially for neuropsychiatric disorders, it's very interesting to see, to use these kind of techniques. And if you're more into research, then you can do subtyping, because of course all diseases are heterogeneous, and connectivity gives you a very good way of separating subtypes of the disease that would otherwise be mixed together. That's fine, but more interesting is prognosis. And that's, I give you an image, I give you some connectivity graph, whatever the modality, and I ask you to predict 6 months down the line, or 10 months down the line, or 2 years down the line, is this person going to be clinically worse or better? I ask you to predict the score. And this is a very interesting application. And specifically, it's interesting for the pharma industry as well, because you can do something called trial enrichment. That is, you can actually try to decide who is more at risk based on this connectivity phenotype, and then you can put them in different arms of the trial. So this is actually an interesting for large-scale clinical research. And the most interesting thing, I think, is this idea of treatment planning, and specifically for things like depression, where if you can know immediately what drug to give to the patient, then you can save lots of quality of life. And it's the same for multiple sclerosis. Multiple sclerosis, you have to try, typically you will try, let's say, interferon, and then within this, maybe it works, maybe it doesn't work, maybe you switch to another class of medications. So if you can predict this from the beginning, I think that's one of the top applications for machine learning in general. And that's not only with connectivity, that's with other modalities as well. But you need large data sets. So hopefully by now you're convinced that connectivity is something useful for clinical neuroscience, but how do you work with this data? Because it's not exactly the same. In fact, it's quite different from the data we've seen. And before doing that, we need to talk a little bit about how you extract this data. So this is just, I want to spend maybe 30 seconds on this slide, going from fMRI to connectivity matrix. If you have diffusion data, it's pretty clear. You do tractography, then you have a connectome. If you have functional MRI, typically what you do is you will have an atlas. You average the temporal signal in this atlas, and then you compute temporal correlations. So that's one way of doing it. There's one thing in here which is very important, is this denoising part, because functional connectivity matrices are very sensitive to motion. Motion is still a big killer for us. We know more and more how to handle this, especially in clinical populations. If you imagine ADHD kids versus control kids, then you will have, of course, lots of motion differences. So this is an interesting one, and also the fact, how do you handle sight differences? It's even worse actually in connectivity data, in functional MRI data than in structural MRI data. Okay? And there's a bunch of ways to do this. So if you're interested, you can go on my site. We have some code for this. Also, you can use the Con toolbox. For R, you can use the package from Sophie Achard, Brainwaver. And in Python, you have some nice stuff that you've seen from Bertrand before. In NiLearn, you have lots of ways of estimating connectivity. So this is very well. Now you don't. It's at this point that maybe if you're not specially interested in this, I would not recommend writing your own package. You can use something else, and that's fairly stable. Now let's talk a little bit about what I mean by brain graphs. So let's say we have one slice of the brain. We have an atlas. We divided the brain into 6 parts, numbered 1 through 6. And this is an axial slice. And each of these dots is a voxel. So that's image space. So now how do you go to a graph? Well, one thing you could do, the classical thing to do, is to say that each of these regions of your atlas corresponds one-to-one to a vertex in your graph. So vertex or node are the same thing. Okay? So we'll play, you see that I have 6 vertices here, numbered 1 to 6, corresponding one-to-one with a brain space. So these are vertices or node, and they're connected by edges. You can also call them links. And the edge encodes the strength of the relationship. If you have functional connectivity, that would be they are consuming oxygen at the same rate, right? So it's basically low-frequency, bold fluctuations that are synchronized. If you have DTI data, then this is basically your tractography, right? You can do this also with morphological graphs if you want. You can be interested potentially in subgraphs. So if you're interested in functional networks, that functional networks in the brain map directly to subgraphs in your functional connectivity graph, or you can call them clusters or communities. And if you have this view, so you can analyze your graph at different scales. You can analyze at the whole graph level, and you can analyze at the community level. And the tool that you will use to analyze your data depends completely on the scale of inference you're interested in. So that's one of the big questions. Are you interested in subparts of the graph or in the whole graph? More formally, these are graphs with fixed cardinality vertex sequences because we have a fixed atlas. So the graph is formalized by a 4-tuple. It's V, E, alpha, beta. That's one way of writing it, with a constraint that we always have the same number of vertices for everybody because we use the same atlas for everybody. And also they're in the same order. So vertex 1 for subject 5 is the same as vertex 1 for subject 35. And if you have this fixed correspondence, then you solve basically a very hard problem in discrete mathematics called the vertex correspondence problem or the graph isomorphism problem. So you basically have this property that things are actually not too bad if you have an atlas, right? So that's actually a good thing. And then some other properties. But basically, what we have is a graph that has some labels in the edges. Okay. And here's what you do with this graph. You could do machine learning. That's what this talk is about. I'll come back to that. You could do statistics, but you cannot do the classical statistics. You have to adapt to the graph structure. Or you could do network science, and you can typically be interested in community structures. So these approaches don't live in isolation. You have links between the two. This is somewhat arbitrary distinction. Nevertheless, I think it's useful conceptually. So in this talk, we're going to focus specifically on 3 types of machine learning approaches that you can use to learn with connectivity data. First is embeddings. Second is graph kernels. And the third is graph-specific neural nets. Let's start with graph embeddings. there are two classes of graph embeddings. One is fixed, you basically apply the same function everywhere, and the other one is data-driven or trained, and in machine learning, this kind of things is called representational learning. At the bottom of the slide, I've tried to mention one researcher which I think is worth following. This one on this particular technique is Yuri Leskovec at Stanford, so I've put also people that are not in neuroimaging because they're doing lots of very interesting things, and perhaps you don't know this person. And they have some very nice code. There's one specifically not by Yuri, but by other people that implements lots of graph embeddings. So you can basically take this toolbox and use it on your own data. What is a graph embedding? Well, a graph, lives in a different space. It's not something that you can just plot in 2 or 3 dimensions. Okay, a graph is its own mathematical object. So you cannot just take your graph and feed it to your SVM. If you want to do that, you have to do an embedding, which means that you're going to find a vector space representation of your graph. So you're going to map somehow, with some procedure, your graph into a vector. Okay? And typically, what you're more interested in if you have a brain atlas is not the nodes so much, but the edges. So the edges have the most interesting information, right? Because they show the relationship between parts. When we're dealing with graphs, we're not necessarily interested in what's happening in parts of the brain. We're interested in the relationships between parts of the brain. Okay. And once you have a vector, you can do whatever you want. So let's see. The first embedding is direct embedding, aka stupid embedding. And what you do with this is you basically take the triangular, you remember that we have this connectivity matrix, right? Region by region, and it's a covariance, and you can do a correlation. You remove the diagonal, it becomes an adjacency matrix. You have correlation matrix minus the diagonal. And what you do is, because it's symmetric, if it's an undirected graph, then you take the upper triangular part and you make it a big vector. Okay, so why did I call this stupid? Because this scales badly, because it scales quadratically in the number of regions. Right? If you have 10 regions, you're going to have in the order of 100 connections. If you have the AL Atlas, 90 regions minus the cerebellum, then you will have 4,005 regions. Dimensions in your vector space. Okay? So you see that if you want to use now this high, nice resolution, 260, the brain netome and this kind of things, 264, then you have in the order of 264 to the power of 2. And that's not a very nice place to be in. You remember this thing about dimensions and data size. So this is not fantastic. So it's a bad representation for this. It's also bad because you lose the relationship between the parts of the edges. And this structure, if you have a correlation matrix or covariance matrix, it has a very nice structure that is positive definite or positive semi-definite, which means that it actually lives in a Riemannian manifold. And so this thing is not a good idea. You could do instead, what Bertrand didn't show in his talk is something they came up with Gaël Varocco also. It's called tangent embedding. It's a much better way. Nevertheless, this is simple and it works most of the time. You don't need any code, just vectorize and you can try it, right? So try it, but be aware that this is really like you have to beat this number. This is like your lowest common denominator between all your methods. So these are some references, and also like if you want to do the tangent embedding, it's directly in NiLearn as well. You can do it. So here's an application of this direct embedding. This is survival prediction for ALS. And what they did here is they had clinical characteristics, structural connectivity, and then some brain morphology. Focus on the column in the middle. And so this is in a sense a connectivity matrix from DTI, so this is from tractography. What they do is they symmetrize it, they take the upper triangular part, and then they feed it to a neural network, right? So exactly what I was saying. So this is direct embedding. They have this connectivity, they make it a big vector, and they feed it to a neural network classifier. And what they show essentially here is that this actually, it actually works, and they can have actually significant prediction of who is going to survive in the short, medium, or long term for ALS. Okay, so it's not a very easy task, but it does work. And my guess is actually if you did a better embedding, you would actually get much better results for this. Another prediction, actually, this is a very impressive paper, I think, I recommend it, from Nature Medicine, and this is trying to predict the treatment response to depression. So this is what I think the most important use case for this kind of techniques. And here what they have is they have a training set, also reasonably sized study with 300 subjects and depressed and 378 healthy, and a replication. So it's a really, I think, really crisp study. what they start with is, again, this embedding. So they have some connectivity data, functional connectivity data. They make this embedding, this big embedding with a direct embedding. And then they do CCA, right? Canonical correlation analysis. This is a way of relating clinical data with connectivity data. You will have a whole talk on this on partial least squares by Valeria, Dr. Kebetz this afternoon, a bit later. And you will see PLS and CCA are the same with some constraints. So essentially what they do is they find subtypes of this disease that are linking, they're basically loading connectivity onto the clinical scores, and that gives them some subtypes. And from this, they can then predict the response to transcranial magnetic stimulation. And it works very well. So this is purely data-driven subtyping from some transformation of the direct embedding of the matrix. At the basis is still the direct embedding. So again, potentially is something that could be improved with better embeddings. Another thing you could do is topological properties embedding. And so here what you could do is, if you know about this clustering coefficient, path length, etc., well, you could extract these nodal properties at each of your nodes of your graph corresponding to a brain region. You could have, let's say, the average clustering coefficient for around the region, and you make that a vector. Right? So now it's a much lower dimensionality because this is in the order of nodes. If you have, let's say, 10 regions in your brain and one graph property, it's 10. If you have two graph properties, 20, but it scales much better. Right? So then you can actually use this to have some clever feature extraction, still not learned representations, but this tends to work reasonably well as well. Right? And then the interpretation becomes a bit more difficult because then you have to say, well, the difference between my patients and my controls is that the clustering coefficient of the left insula is like this, so it's a bit more difficult to translate into clinic. Nevertheless, that can give good results. And it's basically a mix between more clever embeddings and simple embeddings. And then the last technique I want to discuss with embeddings is node to vec. So for those of you that are into machine learning, this is a technique based on word to vec, which is a way of embedding sequences into vector spaces. So this is very similar. The idea of this is that now you're going to embed nodes into vector spaces. It's not about the whole graph, it's about the vertex, right? So take each vertex and you make it into a vector. And the idea is that the representation you find for your vertex, that is, the embedding you find for your vertex, should be similar to the relationship in terms of proximity. So if I have two vectors that are close in vector space, they should also be close in some sense in a graph. And so you have to define a measure of similarity on graphs. One way to do this is you could just say, well, if I start a random walk from this vertex, and I often end up in this, then I have a high probability that these two are related, right? So you can encode this kind of structural connectivity in a graph, this graph structure. And now what you try to find is a vector such that you try to find vectors such that if they're close in the vector space, they're also close in the graph, right? So this is in terms of the path, and this is in terms of vector space. And you do this for all the vertices. So now you're embedding, let's say typically the dimension is about 100. So now you're going to have, let's say, 90 regions of the AL times 100. So you have each of these vectors, and then you have vectors. You do whatever you want. Right? You can do all these machine learning techniques we've seen. Okay. So this, I don't think I've seen an application of this in neuroscience. I think it's, and this is kind of old now, so there's things that outperform. But nevertheless, this is an interesting technique to consider for neuroscience. That was the first technique, embeddings. Now, the other thing you could do is you could also say, well, I don't want to define a vector space. I know what similar graphs look like. And I'm going to encode my knowledge about what similar graphs look like into a function. So I'm going to build a kernel function. If I give it two graphs, this value output by this function will be high if they're similar, and it will be low if they're dissimilar. So a graph kernel is a kernel function, like any other kernel function that we've seen in linear SVMs, for example, except that it operates on graphs. Okay? So this I will skip. Basically, it means that the kernel trick we can do a vector space also works on graphs. And I want to show you one graph kernel that works reasonably well, called the Weisfeller-Lehmann subtree kernel. And let's start from the top left. So this is your original graph, G, and another one, G prime. So the first iteration, you're going to create nodes with an identity that depends on their connectivity. Let's look at the first one. So the first one, you see that it has 5, 2, 3, 4. So you see that now the label of the node is actually the label of the nodes it's connected to. You see? So we went from 5 to 5,235. And we label everything. Then we count the labels, the new labels, and we create new labels to merge them. So the bottom right, the step D here is after relabeling. And now, what you can do is you can build a dictionary of the frequency of each of these labels, the old labels and the new labels. And that gives you, again, a vector, which then you can compute some similarity between two vectors, right? But this is done internally to the kernel. And so this kernel says that two graphs are similar if they have the same subgraphs in there, which is a reasonable thing to say. Okay? So this works reasonably well in practice, and it's been used for brain decoding. It's also been used for clinical applications. So this is a paper from Fay et al. At PRNI 2014, where what they have is they have MCI versus healthy control. So it's a small study, but nevertheless promising results. And here what they do is they use this subtree kernel to actually classify various versions of the thresholded connectivity matrices. So they have a bunch of connectivity matrices, they threshold them, and then they use this kernel, this graph kernel, to do their classification. And they get something like 89, 97% correct accuracy, of course, with huge confidence intervals due to the small sample size. But nevertheless, that's one of the nice applications, I think, of this technique. So this works actually on clinical data as well. And you see that it's really a graph kernel that comes from the general computer vision or even computer science literature. I skipped over this when I went over the slide, but one person to watch in this area is Karsten Borgward from ETHZ. Actually, I should show this maybe a bit longer. So Karsten Borgward from ETHZ, he's got a computational biology lab, and he has this actually toolkit that is called graph kernels. It's also a Python package and an R package, so you can use it directly in your code, and that will give you similarities for your graphs. Okay. And then, of course, the last thing you can do another things, but one thing you could do is you could have a neural network. But neural networks, they like images, 2D, regular fields. They like vectors sometimes, but they don't necessarily like graphs. So this is an area of research that's been very active in the past few years. In the NIPS community. One person to watch here is actually at EPFL, Pierre van der Geijns, but also here, right here in Singapore, Xavier Bresson, who originally was from EPFL, and he's got some nice code in there. So they have some nice tricks to make graphs, convolutional neural nets work with graphs as well in reasonable time. So it's something quite efficient. So here I just want to show one. Maybe you saw this paper because this is maybe closer to this community with a NeuroImage. And so this is from Kawahara et al. So they call this the BrainNet CNN. And what it is a series of filters for graphs that give you, in the end, a decision. So the filter are called edge-to-edge filters. So you give them edges, they give you edges, but filtered, modified by some weights. And they also have edge-to-node filters, which actually correspond, which will give you a single node. They will compress the information. So these are equivalent in the graph domain of convolution in a spatial domain. The key, of course, is that if you have an image, a 2D image, a neighborhood is well defined. If I have a pixel, the next pixel is its neighbor. Just the spatial distance is the same. But on a graph, a neighborhood is not defined by proximity. There's no space, right? The neighborhood in a graph is defined by connectivity, right? It's not the order of the rows of the adjacency matrix. It's the entries of the adjacency matrix. And so what this does is actually encode this notion of neighborhood derived from connectivity to give you predictions. And so they had some nice results for predicting infant motor scores at 18 months from birth. And it's the best result gives you something like 31% correlation, which is 10% variance explained in the score. So it's not very high, but it's a very, very hard task. And they tried also this direct embedding, and this worked much better. So this is a technique I think is promising and should be followed if you have this kind of application. So to summarize the different techniques, so first embeddings, they work reasonably well on several data sets. It's easy to map the discrimination pattern. So you have edge, basically your weights of your classifier are going to be in the edges. The problem is with the direct embedding, you have very high dimensional representation. Graph kernels, they work very well. You can define your own similarity measure, and they're well suited if you have multimodal problems. They're well suited for large graphs, so you don't have a problem with dimensionality. The downside is that generic graph kernel, like the one I showed you, Weisfelder-Lehmann kernel, might not work very well because we have a fixed atlas. And these kernels, they're strong when the nodes don't correspond, but in our case, the nodes correspond. So it might be a downside, actually, for these typical techniques. And neural nets, you don't need to design your kernel or your embedding. You just design the architecture, so you switch your design somewhere else. Works probably well. Problem is, potentially, you have lots of data requirements. Right? And so it means that you typically need more data because they have more parameters. So some in convolutional neural nets with images, typically you can train models with 100 million or 150 million parameters. And in medical imaging, especially clinical data sets in real live hospitals, we don't have that much. And so that might be a problem. So how do you choose between them? Well, it depends if you're interested in a subgraph or in the whole graph. If you have much data, if you don't have much data. So that really depends on the situation and have different pros and cons. So maybe I'll go over by 1 minute if my chairman agrees. Yes. Thank you, Chairman. I just want to address some of the challenges that are, because I'm presenting this, I'm showing you nice results, but you should be aware that this is not, it's not like a done deal. It's actually still difficult. First, motion. So we know that it's the same, if you're doing activation studies with fMRI, with SPM, then you know already that this is a problem. But this is worth, because now you're taking, essentially, if you're taking 2 time series, if you're doing person correlation, you're actually taking the square of these two, so any noise will be magnified. So you can have explosions in your correlation. Just if you have like 1 spike of motion, that's enough to correlate completely uncorrelated data. So motion is still a problem, especially for connectivity. Annotated data is still small. This is like data, we hear all about this, but functional MRI, like structural MRI, is living in relative luxury compared to functional MRI. So in the hospital where I am, it's very rare. It's still a research thing to do functional MRI. Not many people do it, except for preoperative planning, maybe. Although things like UK Biobank are changing the game completely, because now you can have 20,000+ subjects directly with also functional imaging. It's not directly clinical, but the big question, of course, if you do this, and this is not only for connectivity, but it's how to decide between sensitivity and specificity. So is it better to have false positives or to have false negatives? And the calculus is completely different depending on the disease. So for stroke, it's probably okay to have a few false positives because you really want not, like if you have a false negative, the person dies. Right? In other diseases that are maybe slower evolving, like MS, maybe, or maybe it's the opposite. I don't know. This is really, I think, a very difficult question because the same algorithms can be tuned really all the way towards high sensitivity or all the way towards high specificity. And this is something to be aware of, and that's very difficult. And of course, the main difficulty is to integrate data across sites. So most of the theory you've seen this morning from Carsten talk, that you had this idea of IID data, independent and identically distributed, while most of the theory is built for that, but the data is not like this. On this point, I want just to show you, I want to abuse a little bit the privilege that I was granted by my dear chairman, just to mention that most of the large clinical data sets are multi-site. So this is a few open data sets that you probably know, ADHD 200, ABIDE, ADNIGO, and FBIRN. And on the right, you can see the number of sites. Right? So this is actually, you can see like ADNIGO is 50 sites and ABIDE is 16 sites. So we've seen some results in the previous talk from Ninon that actually it might be okay if you train on large data. You might have this cross-site thing, but for fMRI, remember, we're in a poorer situation than in structural MRI. And so this functional imaging data is very sensitive to the acquisition side. So despite effort, so you have with this FBIRN consortium, they have a lot of effort on standardization. So despite these efforts, you still have differences. I want to show you one example of what we did, and then have some conclusions, and it's done. It's about this edge weight distribution. So what we did here on the left is for ABIDE. We have 5 sites at ABIDE. And ABIDE is autism, are denoted in red here, and then typically developing children in blue. And on the vertical axis is the strength of the correlation. So I've just computed the correlation, resting state correlations, and I've plotted them on this vertical axis. And you can see that the distributions are very different. So let's take the site in the middle. Okay. So Trinity. Okay. So in Trinity, you see that the red is much more towards the top. So what if I see this, if I'm at the Trinity site, I'm going to say, well, ADHD kids, yeah, of course, or autism, sorry, kids, they're moving all the time, so of course they have high connectivity, or maybe they have something else in the brain that makes high connectivity. But then if you're on the next side on the right, then you're going to see that the blue, the controls are higher. And so if you are at this side, say, yeah, they have typical connectivity, very strong connectivity, whereas the hypoconnectivity. So you're going to make a whole theory on this, whereas actually it's just a site that's different. I mean, it's also possible that the population, the subjects are different between these sites, of course, right? But I'm going to spare you this, but this FBurn, they have some very nice traveling subject data, so the same subject that went to different sites, and you still see this effect. Okay? So this distribution, that's what we're talking about. When I'm talking about site differences, it's still a major confound. So, to conclude. If you have connectivity data, you can do very powerful single subject inference with, I think, clinical usefulness. You have the methods and the code available. Also, more and more people are getting trained in these methods. In your institution, definitely you can find people. People are interested in collaborating. You should pre-process very carefully. Remember, motion is always your enemy, but also site differences. And of course, the algorithm, I've shown you 3 main classes of algorithms, embeddings. Also, you have many differences, graph kernels and neural nets. These have different goals, they have different data requirements, they don't necessarily look at the same spatial scale, and so you need to look at that. Also, maybe you don't have like a big computer to train these big models, so you need to be careful about that. Here are some useful resources for further study, and I think that's it. Q&A?
Yeah, perfect.
Yeah, thank you. So before the quiz, maybe some questions or no? I don't know.